Glad you're here. Now, are you happy with your cell provider? Everything cells today, figuring out where they came from, what cell theory is, what makes them special, and we're going to talk about some sizes. So, I don't know about you, but I'm going to pull my hair back. And I'm going to check my nails. Students, what you doing? We're going to talk about cells. So let's go. Now, you have seen this cell before, and we talked about it. And you've got a little bit of a shape. So we're going to talk about structures, and we're going to talk about their sizes, and we're going to talk about like how they were found out to start with. So just hang tight with me as we go along. We're going to talk about each one of these components. So we got to go back to the beginning though. Okay, so why are they called cells? What's cell theory? And who figured out what you're made of? So let's first look at cells. So in 1665, throwback to my man, Robert Hooke, okay, he used a microscope to examine thin slices of cork. So those dead plant walls. So if you'll notice right up here, right, all of these, we can count each one of those little compartments, and those are going to be cells. I don't know about you all, but if I use this type of microscope down here, I don't know if I could have seen anything at all. So word up to my man that he could do that. Now, what he saw here kind of looked like little small boxes. Now, some of those boxes look like they are kind of shoved up on top of each other and mushed. However, we look at that and know that it still looks like those small boxes. So cells, because of the fact to him it looked like those small rooms that monks in monasteries live in. They're called cells, not necessarily rooms. And this was a Latin term, cellula. All right, now we're going to jump from 1665 to 1673. Now, the Dutch microscope maker, Ludenvolk, was the first to view an actual living organism. So he used that simple, again, I'm not sure how in the world that you could use that microscope, but down there in the corner, he used his little simple handheld microscope to view pond water and scrapings off his teeth. I don't know about you, but I don't go around like trying to scratch my teeth and go, hey, what's living on those? That's not cool. Ugh. Now, we jump from the 1600s and we're going to go all the way to the 1800s. So in 1838, a German botanist by the name of Matthias Schloden concluded that all plants were made out of cells. So he is considered one of the co-founders of cell theory. And the next year in 1839, the German zoologist Theodor Schwann concluded that all animals were made out of cells. Therefore, he is also one of the co-founders for cell theory. We got our plant man, we got our animal man. So we jump to 18, jumping way too far ahead on our timeline. In 1855, you have the German medical doctor Rudolf Virchow, who are a new type of microscope as they are getting new technologies that cells divide. And so he put the theory to reason that all cells have to come from pre-existing cells, hence cell division. So right here, we go forward and we're looking at those mitotic events. So cell theory, there's three parts. All living cells are made of cells. I can agree with that, right? I'm living and I come from other cells. Cells are a basic unit of structure and function in an organism. One single cell is the smallest thing that I can break down my complete body to, and I can put them in all those individual little cells. The next and the third part, our cells come from a reproduction of existing cells. Now, we're looking at mitosis and meiosis, which is not a topic for discussion today. However, this is what they are talking about. So what's happened since those first discoveries. So endosymbiotic theory, jumping all the way to the 1970s, an American biologist by the name of Lynn Margulis provided evidence that organelles within cells at one time were free living. So they had organisms, or these organelles that we find in today's cells, that seem to have been free living themselves, freestanding, meaning that they had their own DNA. And the two that we talk about here are chloroplast and mitochondria. We will come back to those. They have their own set of DNA, like the rest of your cells in the nucleus. So this shows kind of a little, like, if you want to look through it, that the DNA on those, right, plasma membrane, you've got your DNA cytoplasm here, and you've got that infolding and infolding and infolding and infolding, that you've kind of got this making of the cell. And as it grows and as it forms and as these compartments start to look, they again differentiate into very, very specific parts. So cells, basic units, I can't see them. As much as I scratched my teeth to try to see what he saw, not going to happen. As much as I can pick at my nail, or I could like shake my head to try to get, I can't get one specific cell and be able to see it unless I use a microscope. So there's three basic types of cells: animal cell, plant cell, and then a bacteria cell. Those are the three that we're going to talk about. Now, when we talk about number of cells, we went back to cell theory, and all living things are made out of cells. So in this classification, you're looking at two. You can either be unicellular, which means right? Like unicycle, I've got one wheel. So unicellular means I'm composed of one cell and one cell only. Or it can be multicellular. So I'm composed of many cells, and that can organize into whether it's tissues, organ systems, and we'll get into that a little later. All right, so our multicellular organisms, they are going to specialize. So while all of my cells have the same exact blueprint for everything, they are going to specialize and they can take on different shapes, they can take on different functions, they have different jobs. They're going to be found all over the body, but they are not going to function the same way. I don't need them to. So cells in a multicellular organism turn on and off certain genes based on what they are. So here, I'm going to show you three examples. So notice that your muscle cells right here, they're striated and they're really long, right? And I'm looking at those longer ones. Our red blood cells, I'm sorry, wrong color for the moment. Our red blood cells, notice that right there, they're circular and then they're kind of flat in the middle. These actually do not have nuclei, and so they're going to have a very, very short lifespan too. Notice that your cheek cells are very broken. And I don't know about you, but I chew on the side of my lip sometimes. I'm pretty sure that if I need any of the cells, I can kind of scrape those cells and I can see them like that. Now let's go to plants. You've got some specialized cells as well. Okay, so you've got your guard cells, and notice again, these are more elongated, and then you have right around those stomatas and those that are going to open and close. Notice that this is also under microscopes, those xylem cells that you're going to find. And pollen. There's your grain of pollen for those of you that have that, right? Allergy to everything that starts to bloom, that you have that under the microscope. Again, I can see all the yellow out there and cover my cars and all of that, but to try to fleck one little single piece off to see exactly what it looks like, I'm going to have a microscope. My eyes are not that good. So, simple or complex? That's a question, right? So how do we determine that? When we talk about the prokaryotes, pro before. So I'm looking at before nuclei. I'm looking before all of these organelles. These are going to be my first cells. They lack the nucleus and they lack any membrane-bound organelles. Notice that right there, it is membrane-bound organelles. They might have the same organelles in some of those, but they're not going to be membrane-bound, and they are going to serve a little bit different. So this is including these bacteria cells that are the simplest form. They're going to have a single circular chromosome called a plasmid. So here we've got a picture of one. The nucleoid region is going to contain that DNA, those little plasmids. They're surrounded by a cell membrane. Notice that they have a cell wall. Okay, they contain ribosomes, if you will look, right? We've got all our fancy ones that we've got all of that too, right? We have those ribosomes. However, they are not going to have a membrane around them. They do serve the same function because they're there to make proteins. And we'll talk about protein synthesis later on, not in this episode. Jump to eukaryotes. Now we're looking U meaning true. So notice here that if I look between these two pictures, we're looking at this is more circular, so we know that this is an animal cell. You notice too that there's a lot more in this cell. Okay, so now we've got those membrane-bound organelles, and they're going to have lots and lots of purposes and differentiated between. So these classifications, what do I find in here? I find plants, I find animals, I find protists, and I find fungi. So they're more complex. So if I break down a eukaryotic cell, I can break them down into three basic classifications or these structures. I'm going to have a nucleus, right? In most cases, most, not all, most cases, your nucleus is going to be the largest organelle of your cell. You're also going to have a cell membrane, and then you're going to have that cytoplasm with all the organelles in there. So there's a lot of components going on, but I can put them in one of these three categories. So let's dig a little deeper. Now we're looking at the two main types of eukaryotic cells. We're going to break plants and animals down. Now, if I notice right here, just looking at the two of them, okay, I know that both of these pictures are, one's more cartoonish than the other one. You see a little bit, right? Obviously, it's not the same artist. But if you look overall, notice that the plant cell is more square. Again, we're looking at that fact that we go all the way back to Hook. It looked like it was more of a compartment or a cell. Where you're looking at the animal and it's round. So we're looking at some of the things too. We know that plants, they're not as mobile unless I move back and forth, like with the wind, right? And I want to be taller. I'm going to grow up. And so I need more structure when it comes to some of those and very, very more rigid. I don't want all of my cells to be rigid because I know that, like with my white and my red blood cells, I want them to be able to move in between each other. And so it's going to be a smoother movement. So let's dig deeper into these cells and let's view some of these organelles. So the first thing that we find about these organelles is they have to be even smaller than the cell themselves. So when we get to looking at through the microscope, most school microscopes, at least I know the ones that we use, I can see a cell and I can see some of the parts that are larger and are stained. Like I can see the nuclei. However, without a stronger microscope, I'm not going to be able to see some of these other parts. But I know that there are stronger microscopes out there, and we do have pictures of and been able to see these other parts. So I'm starting to look at there's various functions. Okay, what is it that they do that they have the specific job? And they're found in the cytoplasm. The cytoplasm is going to hold everything in place. Some of these may be membrane bound, and some of them may not, depending on their purpose. Now, here's a quick overview of the animal cell organelles. You'll notice that there's a lot of big words here. Okay, you've got the Golgi apparatus. I'm kind of starting on the bottom over here in the corner and working my way around. Sometimes you hear rough ER versus rough endoplasmic reticulum. Now notice that with this whole nucleus, you've got the nucleus, you've got the nucleolus, and then you've got the nuclear envelope. So nuclear envelope can be interchangeable with nuclear membrane. You've also got attached ribosomes and free ribosomes, and they are going to be protein synthesis and help make those proteins for functions with the cell. You have a cell membrane. You've got the mitochondria. You've got also a smooth ER or smooth endoplasmic reticulum. And then we have the centrioles, at least in this. There can be more that are not listed. So notice here, again, we go back into plants, and a lot of these, right? Golgi apparatus, we have already mentioned, ribosomes, smooth ER, rough ER, cytoplasm, nucleus, nucleolus, and mitochondria. But notice here that you've also, in this picture, have got a peroxisome, you've got a chloroplast, and you have a cell wall, which are three things that we did not find in the other one. So one of the big things is structure and function for some of these organelles are going to be very similar. However, plants and animals do have specific parts that the other one might not have or doesn't have completely. And we're going to talk about those. So let's start at the outside of the cell and work our way in. So we have the plasma membrane, the cell membrane, the plasma membrane. It's also what's going to make up the nuclear membrane when we get into the cell and around those. Notice that right here, there's a lot of components. You have the cell. When you look right here, when it's got the cell membrane, you have this little part that's pink, and you've got it labeled that it's a protein. You've got carbohydrate chains. So again, you've got those sugars, and notice that these are all very different. Okay, so you've got these markers that are going to help everything, but all of them are going to be different. You've got a protein channel, and you've got the lipid bilayer. So bi meaning two. Now, this lipid bilayer, we call them phospholipids, and it surrounds the outside. So phospholipids and proteins are going to be our big two, but why do we need a bilayer? Let's go and talk about that. So our phospholipids, we go back, if you remember, in biomolecules. Okay, if you remember biomolecules, and we're talking about these phospholipids and these lipids being fats, their head of it is going to be a glycerol and a phosphate. That glycerol phosphate combination of a head of a phospholipid are going to be hydrophilic, or I like water. Okay, those tails being of those fatty acids do not. So they are hydrophobic. They're a fear of water. They're going to go away from it. So if you look, if I put all of these hydrophilic heads with hydrophobic ends all together and I drop them in the water, it's right, like what's going to go on? So what we end up doing, and you end up seeing in the cell membrane, is it's a bilayer. So all of those heads are going to be facing either outside of the cell or the inside of the cell that is facing near the water, with the tails that are inward toward each other, that they're moving away from the water concentration on both sides, and it holds its shape. However, it's not going to be a rigid shape. Okay, so they're actually going to move laterally, and all of this is going to allow small molecules to move back and forth. So then you get into things like osmosis and diffusion and those, and what is it that can and can't move? Now, if I'm small enough, I can go ahead and enter, and it might be the O2, the CO2, right? Oxygen, carbon dioxide, water, that might be able to go in and out. However, my cells need more than just these. What do I need as far as when those other parts? What happens if I need something bigger? Notice that this will show you that the membranes are constantly moving. Okay, now we can look at it that water actually, with its polarity, right, with its polar attraction, with looking at this can actually help it move as well. But it's going to still, even though it's constantly moving, it's constantly moving together. So the cell is not going to break apart. Let's talk about those proteins. There's two types of proteins that you've seen in here. Okay, these are going to help all of the large molecules that need in or out of the cell. And it helps with those cell recognition. Sorry, tongue-tied for a minute there. All right, so your peripheral proteins, these are going to be attached to the surface, either inner or outer. And those integral proteins are actually embedded all the way through, kind of like a tube slide, right? Like I can get in and I'm going to slide my way on through one way or another. Okay, these are actually going to go through the whole membrane. Now, this would be one of those integral proteins. Notice that it's on the top and the bottom. It goes all the way through. And these glycoproteins, glyco, the same as some of our carbohydrates, right? Glycogen, glucose. So we're looking at here that these markers to recognize each other or recognize what needs to go in and out are going to be carbohydrate tails. And that's going to help them with the cell recognition. So let's go into plants. When you first go into the cell membrane of plants, right, it's against the cell wall. It's going to be rigid and it's going to help push to maintain that shape. Now, the cell membrane is not rigid, but it's going to push against that cell wall that is to make sure that it holds its shape all the way through there. So we go through, and the cell wall is found in plants, it's found in fungi, and it's found in bacteria. Made of cellulose and made of peptidoglycan in bacteria. Now, fun fact, cellulose, if you like, is one of the many kinds of carbohydrates that humans, we cannot actually digest. So if you go through and you eat like celery or something and you hear that snap, crunch, right, that you go through and you got the stringy stuff, now I can eat it, but some of those things are not going to be able to be digested by my system. And so for us, it's going to pass through us. Okay, so there are certain types of these biomolecules, as far as you're talking about carbohydrates, that animals cannot digest. Now, in fungi, the cell wall is made of chitin. What's the purpose of the cell wall? So it's found outside of the cell membrane, and it is for protection and support. So if we notice right here, this one actually will show all the way around. It more makes it look like that actual cell, right? That rectangular shape. And that cell membrane is going to be right along here, pushing up against that to make sure that it holds its full structure. It's got, again, this plasma membrane right here on the inside with that cell wall on the outside to help protect it the way that it needs. So let's go back further in. Now we're going into the cytoplasm. Cytoplasm, I think about it like Jell-O. It's kind of like that. It's not solid. However, it's not liquid. It's got to have some kind of like shape to hold everything together, but it wants to be wiggly enough that it's going to be able to, right, move and do what it needs to do and not be rigid and break apart. So we consider it a jelly-like substance that is right inside and takes up the cell right inside of the cell membrane. It provides a medium, and one of the biggest things that happens here are chemical reactions. Whoops. All right. So more on the cytoplasm, right? It also contains, well, I think about it like a fruit salad. You ever have a fruit salad where you have all of those little individual like parts that are suspended in Jell-O, like, right? If you get all those little things, That cytoplasm holds that in there, that like my cherry's not going to touch my grape, they're not going to touch my piece of like pineapple, right? So they've all got their little jobs and all of that. So it keeps it apart. However, it's just enough that I can wiggle my bowl or I can go through it, right? Or I can push my finger into it and like, oh, there it comes, and I can move it. So it's kind of got that whole kind of vibe to it too, that it's there to like help all these organelles be able to work out its own job. And this is one that's found in all cells. Now, the nucleus, which you know that this means that we're still, again, either in a plant or animal cell, because we can't talk about prokaryotes. Those are before nuclei. So we're talking about the brain of the cell, right? We're looking at the brain. It controls the normal activities. This also means that it has the DNA and it's bound again in a nuclear envelope or membrane, the same type double bilayer or the phospholipid bilayer that the cell membrane is made of. Now, in most cells, this is going to be the largest organelle. In animal cells, it is. In plants, sometimes you might have one organelle that's bigger. However, this is usually the largest organelle. Now, each cell has a fixed number of chromosomes that carry our genes. Now, we go into humans, and this is moving forward a little bit, right? And we know that we have 23 pair. We've got 46 chromosomes. Now, we have all of that, and that's what makes us human, right? We've got the 23 pair. And depending on that, we could also have an extra copy of one of those. And it would be like, okay, I've got 47 chromosomes, but it doesn't mean that I got a 24th pair. Okay, so I'm looking at 64. There's a type of fern that has 64. There's also a type of fern that has over 100 and something. But we all start looking at different things, and that fixed number of chromosomes that carry those genes make it whatever living organism that we're talking about. Dog, cat, fish, human, fern, Venus flytrap, oak tree. We're all going to have DNA. We're all going to have genes, but we're all going to have chromosomes, but we all have a specific number that is not going to match. Okay? Our genes are going to control the cell characteristics. Do I have white bark? Do I have hair? Do I have fur? Do I have 4 legs or 2? Am I big or small? Right? Not necessarily does the number of chromosomes make me more complex. However, it is going to control what I look like. So now I'm looking specifically at the nuclear membrane, again, talking about that double membrane, and notice that there's little pores. Okay, instead of actually having the embedded protein, you have little pores because there's going to be this little thing called DNA, right, that needs to get those instructions out to make proteins and those other things. And so it's got a specific way of, I can't leave, I'm too big. But if I write that small part of code, I can send it through that nuclear pore to make sure that whatever needs to happen, my instructions are going to get where they're supposed to. Okay, so that is like that opening and closing. But think about it like this. Instead of having a big door that I'm going to be able to go through and I just go bust out through my door, think about it like then I've got a little mouse hole. And so instead of being able to go through the door, I'm going to have to find very small parts that I can actually get to something that can fit through the little door to get what it needs. Now, these nuclear pores, as it leaves the nucleus, this is connected to the rough ER or the endoplasmic reticulum. Before we get there, however, let's go into the nucleus. Okay, so you have that chromatin, and chromatin is going to be where it kind of all looks like it's right jumbled up on everything. And as it starts condense down when we need it to, you're actually going to see those chromosomes. That is made of that genetic material, DNA, which is that fourth biomolecule that we talk about. Going back just a little bit more about the chromatin, right? That DNA spread out, non-dividing. Notice that again, when we start looking at that, they're going to divide if they get too big, and we're going to talk about that before mitosis or meiosis, right? That is going to be the chromatin and non-dividing. As it starts to get ready to divide, it's going to condense and wrap to get ready for cell division. So what does my DNA do? What'd your DNA do? All right, that is all of the hereditary material of a cell, those A, T, C's, and G's. Now, all of these A, T, C's, and G's in this certain point are made of DNA, but I take these little chunks of DNA and I'm going to have those genes. I've got gene for eye color. I've got a gene for hair color. I've got a gene for whether my earlobes are attached or not. I have a gene whether I have, 10 toes or something happened and I might have had 9 or maybe I have 11. You don't know and you're not going to see my feet. Right? So you've got all of these little things and that makes, it also makes us the melanin in our skin. It also is whether or not I'm going to have hair or not, or whether I'm going to lose my hair, depending on. And it gives us other characteristics. It also gives us other characteristics like inside, whether I'm like predisposed for something like diabetes, which, yes, I am. Okay, so you've got those things, and those all are going to make up who I am. These genes also make up the molecular code for all of the proteins that my body is going to have to sustain life. Now, also in the nucleus, something called the nucleolus. Now, I can see that dark, and then I see a really, really, really dark spot in the middle of the nucleus, and that's going to be our nucleolus. Now, cells can have 1 to 3 of those. They disappear when the cell divides, and what it does is it helps to make those ribosomes that are going to be important when making proteins. Ooh, cytoskeleton. Woo! Just as I have a skeleton on the outside and you think about it, right? I've got all of these parts and it's holding me together. I am just not like a Gumby, or I'm just not like a pulled mess on the floor trying to figure out my way around. Our cells actually have to maintain their shape. I do want them flexible and I want them to be able to move. However, I need them also not to collapse on each other. So we've got the cytoplasm and that's going to help, but the cytoskeleton also helps to maintain that shape. Okay, it helps the organelles to move around where they need to be without having a jumbled mess or not enough room. Now, the cytoskeleton is going to be made of proteins. We talk about proteins a lot. We need proteins for this, and I inhale proteins, right? When I eat certain foods and all of that. My body makes proteins, and this is made of proteins, but then I have to build these proteins for this. So if you can't tell, proteins are really, really, really important, okay? So now, going back to the cytoskeleton, those microfilaments are thread-like. They're made of actin. Microtubules, so you're looking at these microfilaments and you're looking at these microtubules, and they're going to be these parts that are going to help maintain, okay? So one is going to be tube-like and made of tubulin, and one is going to be more thread-like. Now, you've got the threads here, if you can tell, and then you've got the tubes. Again, both of those are going to be important to hold that cell together. Here's a different look at those tubules and those microfilaments. Again, notice that micro is on the both of those because, again, they are going to be very, very, very small. Centrioles. These centrioles are actually going to be found in animal cells only. They are paired structures, and they are going to be important when we talk about mitosis. Okay, so they are those microtubules. Notice that they are tubes, right? They're hollow. You've got this, and they are going to form mitotic spindle. So that's going to be those microfilaments that are actually there to pull those chromosomes apart to make sure that when the cell divides, half of everything goes in, depending on whether you're talking about mitosis or meiosis again. And it's going to pull to make sure that that cell divides correctly. And there's parts in both of the new cells. Now, this shows a little bit more of those made. Here, you've got those asters that are going to be made of those microtubules or that tubulin. You've got all of these tubules that are coming out. Okay, they're attaching to those. You've got the little centromeres. About lost my word there. The centromeres on that. So you've got the kinetochore microtubules that are connecting, and they're going to pull apart. You see those centrioles right here, and then you've got the mitotic center. So they're pulling apart, and this is going to pull this one this way, and this is going to pull this one this way as they go through to the poles, and then you're going to end up seeing those split. Yay! Here we go. What everybody remembers, right? Mitochondrion. Plural, more than one. Mitochondria. What are them? Powerhouse, right? Let me see you flex. Powerhouse of the cell. Everybody knows that. Go ask anybody. I'm pretty sure your mom and your dad remember that, right? I'm pretty sure that when we all get there and somebody would go, hey, what's the powerhouse of the cell? You'd be like, mitochondria. That's going to be the Jeopardy question that everybody's going to know, right? But the question is what does it do and why is it important? I get that it's a powerhouse, but what makes it the powerhouse? So we're looking at the fact that our cells, aka our whole body, right? When we all put all these cells together, generate cellular energy. We need ATP. Now, more active the cell is, the more mitochondria is in that type of cell. So I look at it, my muscle cells, my heart, my brain, right? What I'm going to use to flex my arms or like kick my leg, those are going to have more mitochondria because they need more energy. Plants and animals, get this, I'm going to repeat it again. Plants and animals have mitochondria. So when we talk later about photosynthesis and cellular respiration, the site of cellular respiration is mitochondria. Now, I'm going to say that again. Okay? Know that that's important. Both plants and animals have mitochondria, and if the mitochondria is the site of cellular respiration, that means that both plants and animals go through cellular respiration. They do both use energy. So it has a double membrane. You notice that it looks kind of like, I think it kind of looks like a kidney bean there. It kind of looks like a kidney bean. And you've got, when you start to break it apart, right? I've got the inner membrane and I've got the outer membrane. The outer membrane looks like it's more smooth all the way. And the inner membrane looks like somebody took something and shoved it all in, was like, I'm just going to make sure. And it's got all of these folds and sways in it. You've got the matrix. And then you've got the cristae that are through. The folded membranes, the cristae, increases the surface area, and that's going to help more for the chemical reactions to make ATP. All right, so interesting fact, mitochondria comes from the cytoplasm in the egg during fertilization. Ooh, therefore, dun dun dun, you inherit your mitochondria from your mother. So your mitochondria actually has maternal DNA. Back to the powerhouse. Woo, get it done, right? You've got the rod shape. And notice that, again, it kind of looks like that kidney bean part. So why do we call it a powerhouse yet again? It's going to burn glucose to make ATP. Notice that I've got to have ATP again for everything in my body to function properly. On to the ER. Whether you call it ER or whether you call it endoplasmic reticulum does not matter as long as you know what it is. So, what is the ER? The ER is going to be a network of hollow membrane tubules. Okay, it's going to be on one side connected to the nuclear envelope, and it's going to be in the cell membrane. So, synthesis, making. If I'm synthesizing something, I'm making. So, this ER organelle is a synthesis site for cell products and transport. Okay, there's a rough and a smooth. What's the difference? Your rough ER is called rough because it has ribosomes. So technically, I could call it ribosome endoplasmic reticulum if I need to remember what the R stands for, right? So if I call it RER and I think about it, rough and ribosomes, that's what makes it a little bit different. So it makes those membrane proteins, and then the proteins are packaged and exported out of the cell. My cells can make proteins, send them out to be used at a different part of my body. Now, the ribosomes, again, on the surface is what makes those proteins. We look here, it's attached to the nuclear envelope. We just kind of sideswiped it there. And you've got all of the little ribosomes all there. Each one of those sites, each one of those ribosomes are going to end up making proteins. It's not like everything's coming together and just making one. Know that each one of those little bumps that make that rough ER rough are making proteins. Right? They're threaded into the interior, and that helps them for modification and transportation purposes. This right here is more diagram, and this is what you would see as a microscope close up on ER. So let's go to the smooth. Now, first off, right, if we're going, okay, rough ERs because of ribosomes, smooth ER is without them. So the smooth ER is going to lack the ribosomes. They're not going to attach. Now, when I start to look here, we're going through, right? We go back into the cell. We've got the nuclear envelope. Hooked to the nuclear envelope is the rough ER, and hooked to the rough ER is the smooth ER. So we're just working our way out. Now, what does the smooth ER do? It makes products. It makes proteins that are going to be used inside of the cell. So the rough ER exports, the smooth ER uses what's for inside. It stays in-house. Now, it makes lipids like steroids. Okay, going back again, steroids are made out of lipids, also known as fats. Okay, it regulates calcium, so like in your muscle cells, and it's going to destroy toxic substances. It helps the liver flush out all of the junk in the body to get rid of everything that can hurt it. So let's look at those little ribosomes that are attached. They're made of proteins and the rRNA. The R, the little r in rRNA stands for ribosome or ribosomal. So these are protein factories. These are the sites of protein synthesis. Right? They're going to join amino acids together to make proteins. Talked about why that process is called that, and we will go through the process later. So the ribosomes, we've talked about the fact that they can be attached, or they also can look like these little clusters that are moving around in the cytoplasm that are going to be unattached. Ooh, guess what? They're called free ribosomes, and if they're attached, ta-da! Attached ribosomes. Boring, right? Now we're going to the Golgi. I think these are cool. Golgi bodies. They look like a bunch of I think they look like a bunch of flattened pancakes that like start pinching off and like doing right their thing. Golgi is like the post office. What do we think of when we go to the post office? We think of the mail goes in and the mail goes out. So that's what you're going to look at. It has a shipping side. So notice that right here, I've got the trans face and I've got the cis face. Now the trans face is going to be the shipping side. That's what's going out. And I have the receiving side, which is what's coming in. So it receives proteins. These are going to end up being the ones that are going to be exported out. So they receive the proteins by the ER. The transport vesicles are now going to modify those proteins, pinch it off, put it in a receptacle that is going to move. Again, I just want to reiterate, to me, right? They do look like a stack of pancakes, whether it's this, or you go again, what it looks like in the cells. It looks like a stack of pancakes. So what did that stack of pancakes got to do? It modifies, stores, and packages molecules, again, coming out from the ER for storage purposes within the cell or to transport if it's being exported outside the cell. Pretty fancy, right? We go back in. Let's look one more time. You've got the nuclear envelope. You got the rough ER. You have the smooth ER that's going to be attached. And then you have your post office, right? You've got your Golgi body. And look at that. Right there, we've got one of those mitochondria peeking out of that side there. Just a short, it's coming through here. Eat rough ER, it pinches, it's going in. I'm going to look at my post office. This one is going to be packaged and it moves outside of the cell. All right, let's move on to these little things known as lysosomes. Lysosomes are our digestive enzyme body, right? What is its job? It's going to break down food, it's going to break down bacteria, it's going to break down worn out cell parts. Now, it's kind of like, again, it's going to digest, but it's also going to break down, it's going to recycle. And so you've also got autolysis, which is a programmed cell death. So cells can get old, cells get replaced, cells get sloughed off. So these are also going to be part of that process. The lyse is when a cell is going to break open and it releases those enzymes, or that lysosome, I'm sorry, breaks open, releases the enzymes to break down and recycle the cell parts that are unusable or old and worn out. It can take in food, phagocytosis. It will come in, and I'm sorry that my little thing is not working in this. If it comes in, the food will come in, and it's like it surrounds it and pushes it into the cell. Okay, so it can do that, and it can also go around where it's going to pinch off and send something out the other way if it needs to get rid of waste, so that it can things come in and things can go out of that organelle. Big daddy of a plant cell. This is a fluid-filled sac that is going to be for storage. In a lot of pictures, especially in the more cartoony versions of a plant cell that you may see, it looks like a big swimming pool. Now, if we go all the way back to when I talked about nuclei, if you remember in the nucleus, I told you that in most cells, the nucleus is the largest organelle. However, notice here that I could put several nuclei in that vacuole. So for plants, that vacuole is going to be huge, and it's going to be considered a central vacuole. It takes up the majority of that cell. In animal cells, we can have small ones or they can be absent. And again, bacterial cells, They do not have vacuoles at all. So what do vacuoles do in plants? They store cell sap. What is that? Okay, it can include sugar, whether it's the cellulose or whether it's the glucose or any other of those, proteins, minerals, lipids, water, salts, any kind of waste products from plants, and enzymes used for cell purposes. Contractile vacuoles. So contractile vacuoles, we're looking at unicellular protists. Again, this one is going to be a paramecium. Okay, it regulates water intake and then it pumps out excess water to maintain homeostasis. If we go back to homeostasis, it's that internal and that external environment that is going to stay where cells can function at their optimal level. Okay, so this contractile vacuole is also going to keep the cell from bursting open because it can't get out what it needs and it's overfilled. Chloroplast. Now, I think these would be pretty cool to have, but there is no way that I can go out and lay all day in the sun and I'm ever going to make my own food. Okay? When we talk about making our food, we are not talking about the fact of, hey, I can go microwave that pizza. Great, good for you. When we're talking about chloroplast and making our own food, we're talking about producers. So we're looking at something that can produce their own food by using the sun and converting it into something else usable. If I go lay out in the sun, I don't know about you, but I go from this to looking like I was cooked lobster, and then I'm going to go back. Okay? I am never going to turn green. I am never going to end up being able to make my own food. So, chloroplasts are found in producers, and they're going to contain the pigment chlorophyll. The chloroplast is the location where photosynthesis is going to happen. Chlorophyll is the pigment that is used. What does it do? It converts solar energy or sunlight into something more usable. In this case, for plants, it's going to be making their food or that glucose. So the energy from the sun is going to be that solar energy made into the chemical energy that is found in those bonds of those sugars. So if I break this down, we're going right back to some of those same things that we talked about in the mitochondria. You've got it as a double membrane. You've got that outer membrane and you've got the inner membrane. The outer is smooth and the inner membrane are called these thylakoids. Now, thylakoids to me, instead of going to the Golgi and saying, hey, they look like little stacks of like pancakes at this time, it almost looks like it's a bunch of nickels that are, it could be really, really small pancakes too, but for sake and purpose that we don't want to get mixed up on that, we're going to look at these. They're little like stacks of pennies or they're little stacks of nickels. Okay, so that inner membrane, again, they're going to be those little sacs called thylakoids. And when I start to, those are each one of these individuals. Think about each one of those nickels are called thylakoids. When I make them in stacks, they're going to be called grana, but they're connected. So some of those, if you've ever seen anybody do it, that it's like they glue everything together and then glue it to the floor and you can't move it. That's kind of what that's going to be. They are going to be put together, but they're going to be put together in a way that can't be broken apart. Okay, they're interconnected. And that stroma is going to be kind of like the cytoplasm. It's the gel that's going to be in the middle surrounding those thylakoids. So here is your other mitochondria contained its own DNA. Chloroplasts also have its own DNA. Now, chloroplasts contain enzymes and pigments for photosynthesis. If I cannot photosynthesize, and again, I cannot take the sun and convert the solar energy into something my body can use, I have to eat, right? Something that has been made. I have to eat plants. I got to eat whatever. So I am not a producer. I do not photosynthesize. So again, it's emphasized this is never in animal or bacteria cells. Again, right? Breaking down, photo meaning light, synthesizing meaning I'm making. So I'm making food from sunlight. Ooh, bonus time. Here we go. Are cells in an elephant? Try it again. Are cells in an elephant bigger, smaller, or about the same size as those in a mouse? Hold that thought, we'll come back. So let's look at factors affecting cell size so we can answer this question. Your surface area, the plasma membrane surface, right? Cell membrane surface. That is determined by multiplying the length and width, or the L times the W, right? And we're looking at the volume, what's in, and it's determined by the length times the width times the height. So I'm looking at that, and we know that length times width, and then length times width times height, volume increases faster than the surface area, or meaning what was inside the cell increases faster than the plasma membrane can get. Okay? So when I look at that, surface area is no longer great enough to get rid of all the waste and to get enough food. So if everything inside gets too big, that cell is going to end up having to figure out what it's going to do. Okay, there's only two options. It either gets so big and it's kind of like a balloon, it gets so big and it goes boom, and we don't want that to happen. Or it's going to have to get to the point where it gets to a place where it can divide. Okay, so the cells of the organisms are going to be close in size. They all get to a certain point, they all got to divide. They all get to a certain point, they all divide. Going back to that question, I gave it. Are the cells in an elephant bigger, smaller, or about the same size as those in a mouse? Looking at what we just concluded, they're about the same size. But the elephant has many, many more cells than that mouse is going to have. So right here, we're going to switch gears a little bit and look at the organization of that life. Now we're looking at cells and we're looking at all the components, and we know that how all this fits together, and we know that cells are needed and all of this craziness, but how does that matter? Okay, so on non-living levels, and we're going through atoms make molecules make the organelles, right? So we know those little pieces. Never trust an atom, right? They make up everything. Ta-ding, psh. Okay, so you put all those multiple things right together and we get the molecules. The molecules are, we're looking at H2Os and CO2s, and you've got C6H12O6s, and you've got all this, and you put all these things together and you get the organelles. So at the living level, we're looking at the fact that you've got cells. Cells going to start life, and you've got multiple different types here, right? We know that this one is going to be that really elongated cell, and it's the ones that are like my nerve cells. And then you've got the striated one here, so that's going to be like my muscle cells, right? So we take cells, and what if we put all of those cells together? What do? So cells, pop them all together, the same sort, and they're going to be tissues. Tissues are going to be similar cells that are working together, right? Got my muscle tissue. I've got my skin tissue, right? I've got all of those different types of tissues. So what am I going to do? I'm going to take those different types of tissues and I'm going to put them together, okay? Now, when I put the different types of tissues working together, those are going to be my organs. So notice here I give you the stomach. The stomach is made out of multiple different types of tissues put together that is going to make that organ. Now, I'm going to put all of those different organs together, right? And in this case, from the moment that something enters my mouth until it comes out the other end, that whole system right there is known as our digestion system. So now these are going to be organ systems, right? Whether it's my digestive system or my respiratory system, talking about my lungs and the other organs that go with that, or my circulatory system, or whatever it might be. I'm looking at those organ systems. Now, I can't live with just a digestive system or just being able to breathe without, like, right, all of these other systems. So I'm going to put all of this together and I get the actual organism itself. Okay, so I get, notice here, I've got that elephant and I've got that fish, little clam looking at you, right? And I've got the dancing fungi over there, right? That's why they party. They're fungi. Dun, dun, shh, there you go. Right? And you've got this tree and you've got the birds. And I didn't put a picture, but I could have put one of me, right? So we make all those organisms. So I really, really hope that this gives you a big or quick or easier overview of exactly what is so cool about cells, right? And why they're needed, why they have everything, why, like in the beginning, that we knew that when cell theory that we had to all come from cells because living things come from other living things. So I really, really am excited that you were here. I really hope that you got a lot of anything. If you need, you know how to get a hold of me. Any questions I can ever have, I hope to see you next time. So, if you got all that right on the same thing now, I'm going to take my hair down, check my nails. Hope you really had a cool time talking about cells. See you next time. I'm out.